Hi guys, um, welcome back. So with this lecture, we are going to talk about the muscle. So we can talk about different kind of thing about the muscle. This one I'm going to focus on the anatomy of the muscle. So we are not going to look into the uh, how does this functional or molecular change in the muscle. We will just focus on the very basic uh, anatomical understanding to get an anatomical understanding of the muscle, right? So let me share my screen. Share. Yeah. All right, good. So that's the slide. And so I have I think probably we'll do like three small video clips to describe this lecture. So the very first one is to just give you an overview of the muscle macroscopic and microscopic features. So our human body has three type of muscle. We talked about this one in our uh, earlier lecture when we talk about the tissue. When we talk about the muscle tissues, we describe there are three type of the muscle. We have the skeletal muscle, cardiac muscle, and the smooth muscle. And from there, we know each muscle has its feature. The feature of the skeletal muscle is that there are probably two or three things. One thing is that skeletal muscle, by the name, you know, it's connect with the skeleton, it's connect with the bone. So other muscle do not connect with the bone. Only this muscle connect with bone. So that's one feature. Another one is that you can see it has a striated appearance. We see this striated appearance in both skeletal muscle and cardiac muscle. These two you find this striated appearance. We later will learn why do these muscles have this striated appearance. It's all because of their the microscopic uh, anatomy have a very unique arrangement of this myofibril. So we will look into that. But just for now, we know that just by looking at this muscle, it has this striated appearance. So that's the second thing. The third thing about the feature, the unique feature of the skeletal muscle is that skeletal muscle has multi-nuclei. So typically, one cell has only single nucleus. Yes, single nucleus. That single nucleus contains the chromosome, contains DNA, contains the genetic coding for these cells to function. But for the skeletal muscle, because the muscle is pretty long, this muscle is pretty fiber is pretty long. Each fiber is a muscle cell. This cell is pretty long. So this cell is produced by merging many cells all together. So because we have multiple cells merged together to become one single cell, and so this finalized cell has multiple nuclei. And another thing that you can also find is that these nuclei do not stay inside of that middle of the cell. It stay in the peripheral um, area of the cell. So the muscle cells pretty much put every space they have to generate muscle movement. And because the space is limited, and so everything else kind of pushing to the border. Yeah, so that's three unique things. We will focus on this one first, then we will also spend time to talk about cardiac muscle and the smooth muscle. All right. So another thing about the skeletal muscle is that it's voluntary control. Cardiac muscle and smooth muscle are not voluntary control. We cannot many specific control our heart muscle. It just operate on its own. We call it an autonomic control. But for the skeletal muscle is not autonomic. It's controlled by our voluntary, like your mind, your thought. your desire. You drive those muscles to move. So when we look at this muscle, skeletal muscle, what we need to know is that this is the muscle that connect with the bone. So what do we see here? So we see muscle organ that connect with the bone. And so we in our lab we have trying to identify different different part of our muscle organs has their names. So we learn those specific names already. But if we look at this muscle, we can see that all these muscle connect with the bone. So here you have one thing, that's muscle, and that is another thing, that's bone. So muscle connect with bone by what? It's connect with bone by tendon. So that's another terminology you need to know, the tendon. Tendon brings muscle, attached muscle to the bone. Yeah, so you need to know the rule of this tendon. Tendon injury is because the muscle exercise cause some tears in the tendon, and this can be repaired just naturally. Our body can repair it by itself. Um, so what is tendon? Tendon meaning that it's a connection of the muscle to the bone. When we see this organ, each muscle organ has a name. And how do we know that is an organ? Because that is a individual muscle. It has two attachments, one attached to one bone, the other end attached to another bone. So this muscle organ has two attachments. Sometimes has two, sometimes more, depends. But they're attached to bone. Now this organ, what we can see is that it has these fiber. Each of these fiber is a muscle cell. So muscle cell is pretty long. Each of these has muscle cells. What we find here is that all these are wrapped by a membrane. So this muscle organ is wrapped by a membrane. There is a name for this membrane. This name is called epimysium. So muscle organ is wrapped by epimysium. When we peel away these epimysium, we can find there are multiple bundles of this fiber structure. And this next level of this bundle, when we tear into it, these bundles are wrapped by another membrane. So this is another compartment. This membrane is called perimysium. So perimysium bundle many muscle fibers all together, and these are longitudinal muscle fiber all together. And this compartment within each perimysium is called fascicle right here. So fascicle. Now, fascicles include many muscle fibers. Each muscle fibers is a muscle cell, and we also have additional membrane to wrap around each muscle fiber. This membrane is called endomysium. And so that's overall, look at this. This is one of these fascicles wrapped by perimysium. And here is single muscle fibers. Each single muscle fiber is a muscle cell. So that is the overall arrangement or organization of muscle of the skeletal muscle. So you can see that skeletal muscle is pretty elongated. It's a muscle fiber, just like when you eat your uh, chicken or your beef or your like dietary meat, you can see that those muscle has the fiber, and that basically represent all this fiber you see here. Each muscle fiber, if you can hear it, each single muscle fiber is a muscle cell, and the multiple muscle cells wrap together to form a fascicles, and these fascicles are wrapped by perimysium, and then multiple vesicles will wrap together to form a muscle organ, and these muscle organs are wrapped by epimysium. So that's the overall structure. And then yeah. So and then the next thing is that okay, when we knowing all this, the next thing is to know looking further detail, looking into more detail into the muscle fiber. So here is, all right, another thing before we move to muscle fiber. Another thing I also want to tell you is that this is the overall structure of this arrangement. Sometimes if our muscle organ is pretty big, if our muscle, you can see that vesicle basically is wrapped, is a compartment that contains the muscle cells or muscle fibers. Between the vesicles, you have the blood vessels, you have the nerve to go through. That's how we nourishing our muscle. Muscle need energy. Blood vessel will bring the energy. Sometimes when the muscle is pretty big, a large muscle, organs, then we will have not just these uh, standard separation or compartment, we have something a little bit more to enforce the structure. And that something more is that some of these vesicles were wrapped separately with another package, And so that's this one here. So here we call it vesicle compartment. So here you have one vesicle, you have another vesicle. Individual vesicle is already wrapped by perimysium. However, here what we see here is that these vesicles, this vesicle, and this vesicle are further grouped together by another layer of the membrane. So that will enforce the structure stability, because not just only one membrane to package it. Now your package is much bigger. You need double membrane, and so this double membrane, the additional membrane to wrap around multiple vesicles together, this membrane is called fascia. Sometimes we also call deep fascia because it's deep into the muscle by fascia. And so what does fascia wrapped? What does fascia package? Fascia package multiple vesicles. So yes, in this case, it packed 3 vesicles. In this case, it wrapped 3 vesicles. In this case, it wrapped 2 vesicles. So each fascia wrapped more than 1 vesicles. And we call this group of the vesicles vesicle compartment. So that's just another layer of the packaging of our muscle. So let's look into the microscopic structure of the muscle cell. So when we just tear it apart and just look at single muscle fiber, we can realize that, okay, this muscle fiber is a muscle cell. Even though it has multiple nuclei, but it has everything that a cells have. So a cell has a cell membrane. So you also have a cell membrane here. All right, so I think it's recording. Just double check. Yes, it's recording. All right. So this cell has everything that a cell has. So it has a nucleus. It just have more than one nucleus. With plural, we call it nuclei. So we have more than one nuclei right here, multi-nuclei, and they just pushed into the border of a cell. You can see that it has a mitochondria, just like every other cell. It has a cell membrane. It's elongated, so it's elongated. And this cell membrane here, it has a name. This cell membrane is called cell membrane right here. It's called sarcolemma. It's just a name. but it's a cell membrane. It's the same thing as other cells have. What we also observe here is that inside of it, it has this protein structure. So inside of this cell, they have proteins. And earlier we learned that, okay, cell has a cytoskeleton. And so it has the muscle cells use those cytoskeleton. If you remember that we have three type of cytoskeleton inside of a cell. We have microtubule, microfilaments, and intermediate filament. So we have three type of cytoskeleton. It's a skeleton means like a skeleton structure, and the cytoskeleton are the one to form the structure of a cell. Yeah. So muscle use that cytoskeleton to do the movement work. We will look into that. And so here is that or arrangement, that organization of this cytoskeleton. So you can see that each of these is called myofibril. And this myofibril is bundle of this protein, including the cytoskeleton. This microfibril, if we zoom in right here, if we zoom in, we can see that this microfibril is these bundles of filaments. So you can see that it has some filaments sit in the middle, and some filaments sit in two ends. So that is this myofibril. What else do we have here? So we have the mitochondria, like I mentioned earlier, sarcolemma, the membrane. Here, myofibril. Myofibril is a bundle of some of these filaments, some of these filaments, and some of these filaments. Microfibril contains two types of filaments. One is called thick filaments. That's the one located in the middle of that segment, middle of it. And we also have thin filaments located in the two ends of these segments. So you can see that these repeat basically. You have the thick filaments. This is the middle line, thin filaments, and this will repeat over and over again. And so with that, if we look at the border, we will find this region is a band, then there is another band, the thing repeat, we have another band. And because of that, we have this striated appearance. Yeah. We also have something called sarcoplasmic reticulum. So this blue thing, this network thing that you can see here is called sarcoplasmic reticulum. What is it? This is basically the same thing as endoplasmic reticulum. So in our typical cells, we have these to help with the protein synthesis. Basically, in the sarcoplasmic reticulum, we have rough one, we have the smooth one. The rough one has the ribosome that is involved in translation to convert RNA to protein. And same thing here, you have the sarcoplasm. It's called sarcoplasmic reticulum in the muscle, but it basically is the same thing as the endoplasmic reticulum in the typical cell. When these are another thing that we can also see here is called T-tubule, right here, T-tubule, because muscle is pretty big, muscle cell is pretty big, and so inside is basically deep away from the peripheral. And this tubule allow some communication from the peripheral get enter into it, like tunnel. This one is like a rapid tunnel in a way. So you can see that this tunnel coming in, and they can basically go into the muscles cell. This one is called T-tubule. T refer to the transverse. And so basically cut through the muscle cell. cut through each cell. And this T-tubule will in a way to separate the filaments into each myofibril. Yeah, so you can see that this one myofibril, and myofibril basically is bundle of thin filaments and thick filaments. The endoplasmic reticulum in the muscle is called sarcoplasmic reticulum. Basically go along with this myofibril, and when they encounter this T-tubule, they will basically just kind of like stop and then appear again. And so this structure is called a triad. It's just the way to describe this interaction or the uh, adjustment of the sarcoplasmic reticulum and the T-tubule. So that's that. What else? The end of the sarcoplasmic reticulum is called terminal cisterna. It's just a terminology. It's the same thing. It's sarcoplasmic reticulum. It's just different location, has some terminology to describe that location. Then here is the sarcolemma. That's a membrane. Sarcoplasma is a cytoplasm. Cytoplasm is inside the cell. Myofibril. So that's that. So that's the structure. What would we know further? What we need to know further is this thing, because this is how muscle conduct the movement. This myofibril. The next thing we are going to look into is looking into the arrangement of this myofibril. Particularly, we're looking into the arrangement of thick filaments and thin filaments. Yeah. So that's that. So this summarize what we talk about. Focus on what is important. One thing you need to know is that each muscle fiber is a muscle cell. Yeah. And each muscle cell has multi-nuclei. I think this is probably the only cell in our body has more than one nucleus in the cell. Maybe there are other cells, but this is the only thing I can think about. Yeah, but that's pretty unique. Yeah, there are some rare situations that you may see multi-nuclei in some other cells, but generally speaking, in a normal situation, skeletal muscle probably the only one I can think about. Yeah. All nuclei are located near the plasma membrane. This plasma membrane is called sarcolemma. Each muscle contains multi-myofibril, and the myofibril has thin filaments and thick filaments. So that's this one here. Myofibril has thin filaments and thick filaments. And the one thing you also need to know is that the thin filaments and the thick filaments is not randomly distributed. They are very well organized. So that's the next thing that we are going to look into. So this is a cartoon showing you the arrangement of thin filaments and thick filaments. What we can see here is that we can put thick filaments here and the thin filaments on two ends. And so by this arrangement, and we can see this arrangement can repeat itself over and over again. And because of this arrangement, because of this repetition, the muscle will have appearance like striated appearance. So this one will be dark, this portion will be light, and then repeated, you will have dark band, light band, dark band, light band over and over again. So that's that. So so this is the thick filaments are in the middle. Their middle the middle of these thick filaments is is uh, is is attached to a line is called M line. M refer to the middle line. So this is middle line, and the thick filaments, the middle of the thick filaments attached to that M line. And we have thin filaments. Thin filaments are in two ends. These thin filaments are attached to Z line. So this is like zigzag. So this is like Z line. Thick filaments also attached to the Z line by a protein. This is called tintin. So this one is an elastic protein. So we will see how it goes. Yeah. So that's that. So that's the arrangement. From here, we can see that this is a unique, is a very basic unit of the muscle myofibril. So this thing will repeat over and over again. And this unit has a name. This unit is called sarcomere. So that's a terminology. This one you got to know. What is a sarcomere? Sarcomere is a unit of the myofibril segment. And the definition of one sarcomere is that the segment between two Z9 is a sarcomere. So that is one sarcomere, and that is another one sarcomere. That is another one. That is another one. So that is the sarcomere. And the sarcomere is a segment between two Z9, and it has everything. And then the next sarcomere basically repeated over and over again. Yeah, so that's that. We also can see that with this arrangement, the sarcomere can have have different bands. So here we have the A band. A band is the one that contains the thick filament. And we also have light I band. I refer to the isotropic, meaning that its compartment is pretty much the same. A band has both thick filaments, hence and a portion of the thin filaments. So that one is not so isotropic. A refer to N isotropic right here. So that's that. So what I forgot to mention, what are the thin filaments and what are the thick filaments? Thin filaments is formed by actin. And remember that actin is the element to form the cytoskeleton. And so that's the same thing. This is cytoskeleton in other cells. And so that's made by actin. Um, Thick filaments is formed by another element that is called myosin. This myosin has this sticky part. It gonna attach to the thin filament to do the movement. Yeah, so just keep that thought in mind. We will talk about that later. And then here's the A band, that is the I band. Yeah, so that's that. So now knowing this, let's look at how do these filaments conduct the movement. So what is the movement? Movement basically is how muscle move our body, right? Move our body, basically move our body parts, not entire body, but probably move the body part. And for the skeletal muscle, when they move, basically what they do is they do the contraction. So just like your finger, right? Your finger can bend. Is because this muscle and this muscle contract. When this muscle contract, let me go back to here. When this muscle contract, it gonna pull a bone, and so your body can move. So that's how our body moves. It's by pulling the bone. And by pulling bone, we basically do the muscle contraction, becomes shortening. Muscle becomes shortening. Yeah. So how does it do it? The way it does it is by sending these thick filaments. These thick filaments has this myosin. This part is called myosin head. This sticky part. So this sticky part gonna attach to the thin filaments. And then once they attach to it, they're gonna pull it. So you can see that these thick filaments attach to it and they pull it. When they pull it, this part gonna moving toward the middle. When they attach to it, pull it, and then this part will moving into the middle. So what it does is that when you do the when it is activated, the thick filaments hook onto the thin filaments and then pull these two Z lines together. So this sarcomere, this band become shortening. This segment becomes shortening. So that's how they do the movement. It's by attaching the thick filaments into thin filaments and then pull it together to the middle. So let me show you this picture here. So this is how they do the movement. So here you have the thick filaments. It has this sticky portion. It attached to the thin filaments and then pull these two things together. Pull this line together. When they pull this line together, the muscle becomes shortened. So that's how they do it. This process is also called sliding filaments process because it's very much your filaments slide through this thick filament, they just slide through it, and then your muscle becomes shortened. So that's that. So you can see that this part is called edge zone, like a hollow, empty. It will get closed. I-band will be shortened a little bit. You can see that A-band pretty much the same because A-band is where we have the thick filaments. Thick filaments, the length is not going to be changed. However, 2Z line will get close together. Yeah, so the overall, the sarcomere will get shortening. So that's how the muscle do the contraction. And so that's it. That's everything that you probably need to know. The remaining of it basically is the continuous from there. The next thing that we need to know is that, okay, that's this one here. Sliding filaments, when muscle contracts, meaning that it becomes shortening, that is when the muscle is doing the work. It becomes shortening, and then the bone is going to get pulled. What it does is that the myosin thick filament, that is myosin, sliding along the actin filament, sliding along the thin filament. The myosin attached to the thin filament, and the two thin filaments sliding through it, approaching to the middle. And that will cause a shortening of sarcomere. Sarcomere is the segments between two Z lines. They will become shortening. And then that will shorten the muscle. This process need to be need energy. You know, all this movement require energy. These sliding filaments require energy, and our body's energy are produced by ATP, so they require ATP. You need to eat. You need to your cell. You need to use oxygen. Need to use glucose. Need to use fat, and then to produce ATP, the organelle to produce ATP is mitochondria. And so you need that to produce ATP. Then ATP is being utilized by the muscle to generate this muscle contraction. So that's that. This is last slide before we take a break. What I show you here is that there is an optimal tension that can be generated by each of these process. So if this sarcomere, this one showing you the x-axis is the length of the sarcomere, the original sarcomere, and the y-axis show you the tension generated by the sliding filaments. If these two sarcomere, if this sarcomere is very short, very narrowing, there is no space for it to contract. So the tension is very little. You cannot regenerate too much of force. If this sarcomere is so long, there is no space that thick filaments uh, can pull it. These thick filaments has this sticky part, but it has nothing to sticky on it, to get stuck on it. And so it doesn't have something to hold on it. It doesn't really generate the force that much. So the optimal length of the sarcomere or the optimal length of your muscle to generate the largest tension or larger force is something between. It's not too long or it's not too short. And that is why when you have the competition, you have people, compete for the running, you want to position yourself that your muscle is not too long, you don't extend it too much, or you don't shortening it too much. Keep it into that optimal position, then it will generate the optimal force, largest force. You can generate larger tension from that optimal length of the sarcomere. And so that is everything that you need to know for the sliding mechanism. And with this short video, we went through from the gross anatomy, knowing about the organization of the muscle organ, down into the organization of the uh, myofibril within each muscle cell. And we understand the thick filaments, thin filament, how do they interact together to generate muscle contraction. So that's that. All right, let's take a break and we will continue in the next one.